Okay, welcome to the final colloquium session for what is this? This is the spring of 2023. We're really excited to extend our colloquium by one presentation this time around, so we can hear from Tim Rhoads, who is a candidate to join our program as a trainer in the genetics PhD program. So Tim got his undergraduate degree at Gonzaga and then did his PhD at Oregon State before coming to UW-Madison as a postdoc to work in two really fantastic labs. I'm embarrassed to say, apparently he worked in the lab right next door to mine for 4 years without us meeting. So in Josh Kuhn's lab, where he honed some skills in proteomics to go with his underlying skills in genetics and genomics. And then after that has been working with Roz Anderson in the VA and focusing on a subject we're going to hear more about today, which is metabolic reprogramming. And specifically what Tim has been working on is something we've all heard a lot about over the years about caloric restriction and aging. And what has been lacking in every presentation I've heard about it is really getting to the heart of the mechanisms behind it. And I think that's one of the really exciting things about Tim's research. And so without any further ado, I will let him tell you about it.
Well, thank you, Nicole, for that very nice introduction. Can everyone hear me okay? I am very honored to get the chance to talk to you all today. And so as Nicole mentioned, I'm sort of brand new starting assistant professor in nutritional sciences. So work in my lab is really just getting going. And so what I'm going to talk about today is kind of the backdrop of data that I have worked on in the past that kind of informs the research direction that my lab will be pursuing, as Nicole alluded to really drive at the mechanisms of caloric restriction and aging. And so the part of it that I'm very interested in is gene expression regulation and how that is involved in metabolic reprogramming. And as you can probably guess, based on the picture that I have on the title slide, I'm going to talk a fair bit about my involvement with the non-human primate calorie restriction study that's been ongoing here at UW-Madison since the late 1980s. So there we go. Okay, so first, really quick, why am I interested in sort of aging metabolism and its relation to disease risk? So this is a schematic from a perspective piece that I wrote with Roz about a paper in Science that describes energy expenditure measured via doubly labeled water. They measured it for thousands of individuals ranging from basically newborns all the way up into individuals in their 90s. And so what they found was rather interesting. So this yellow trace is sort of a depiction of the energy expenditure across the lifespan. It peaks sort of as a child and adolescence, and then settles down around where it's going to be at age 20 for adulthood. And the thing that was interesting about this actually is this sort of flat part here. What a lot of people had assumed was that energy expenditure and metabolism would sort of slowly decline from this point. And so I could blame my expanding waistline, not just from moving to Wisconsin and eating cheese and beer, but just getting older and my metabolism slowing down. It turns out that's not really what happens. And energy expenditure is pretty stable across the lifespan and really only starts to decline once you hit age 60, 65. The thing that we found very interesting about this from an aging perspective is that is right around when the risk or the incidence of chronic diseases really starts to go up. And so advanced age is the largest risk factor for a number of chronic diseases, cancer, Alzheimer's disease, early sarcopenia. And so we don't think that it's a coincidence that right as metabolism starts to kind of decline, you see a really increased risk for these chronic diseases. So that's also one of the reasons why caloric restriction is a really interesting model to use. It was first sort of formally described in the 1930s, experiments in rats. And basically it's a decrease in nutrient intake without malnutrition. And so what you see on the slide here is data from mice. This is reproduced from Rick Weindrich on a couple of different degrees of restriction and showing the corresponding decrease in body weight, and then also the percent enhancement in survival. And if you plot sort of lifespan versus caloric intake, you get this almost inverse linear relationship. And I think that this really highlights that metabolism is kind of the key factor when it comes to overall lifespan. Now, caloric restriction has been studied in a lot of different organisms since it was first described in the 30s, but it was an open question for a long time how well it was going to work in primates. And so to address that, and because it's really hard to do with humans, a couple of non-human primate studies were started, one of which was here at UW-Madison back in the late 80s. And so this was done in rhesus monkeys. So these animals live about 26 or 27 years in captivity. And the study here was starting the animals around young adults, so 8 to 14 years of age. And the target restriction was 30%, and they were maintained on that for their entire adult life. And so that study has published a number of times. And so the picture here is from the 2009 Science paper. Hopefully, it's fairly apparent which animal is ad libitum fed and which one is calorically restricted. I think it's pretty obvious that this guy is absolutely angry enough to be hungry, and is definitely the restricted one. And both of these animals, I should point out, are right at the median lifespan for the species, so 27 years of age. And we're now in 2023, all of the animals in the study are deceased. So we have complete survival curves for the study. So on the left is all-cause mortality, and on the right is age-related mortality, and that's assessed by a blinded pathologist. In both cases, we have a statistically significant benefit in survival to the restricted group. And in the case of the age-related survival, it's quite dramatic. So we think of this as a successful implementation of CR, and this is a model of delayed aging in these non-human primates. So then the question becomes, why? What is it molecularly that is leading to this improvement in longevity outcomes? So this somewhat busy slide tries to summarize the sampling of these animals over time. So the blue and red ticks are the necropsies when animals died. And then we also have longitudinal biometric data, PBMCs, and functional data at different time points. So obviously we'll only be able to scratch the barest of surfaces with the data available. And there were also biopsies collected at a few different time points from a couple of different tissues. And that's primarily where I'm going to focus the part of the talk where I'm talking about the monkeys. We had biopsies collected from liver very early on in the study, and then skeletal muscle biopsies collected from older animals. And so the first thing is to talk about the liver story, just the hepatic response to basically short-term CR. And we did what I call kitchen sink molecular profiling. If there was an omic that we could do, we did it. So RNA-seq, proteomics, acetoproteomics, lipidomics, metabolomics, all done in collaboration with groups here on campus. Roughly 22-ish thousand molecules that were quantified across the two time points. And if we look at the data from these animals, so this is sort of going one ohm at a time. So transcriptional data and looking at a pathway analysis. What we see is sort of an upregulation of metabolism pathways, branched chain amino acid, OXPHOS, TCA cycle, and kind of a downregulation of immune and inflammatory related pathways. And this matches fairly well with what's been observed in other organisms as a response to CR. The thing that was a little bit new to us and less was known about was, so RNA transport and spliceosome, there are a few other RNA-based pathways that are scattered in there that aren't labeled. And there were a couple of recent papers right around when we were putting this together that had described the participation of different splicing factors in the mechanisms of VR. And so we wanted to try and dig into that a little bit here. And so what we did is we used a computational tool called DEXseq, looks at short read RNA-seq data and assesses sort of differential exon usage. And what we found over that 2-year sampling period is that it's really not happening in the control animals, whereas in the CR animals, there were roughly 400-ish genes where we could find at least one exon where there was a change in its usage. And that is indicative of some sort of RNA processing like alternative splicing or potentially an alternative start site. And so this is something that I'm going to come back to a couple of times throughout the talk because this has become a mechanism that I'm very interested in trying to work out what it's doing. If we look at the proteomics data, so we basically observed similar pathways coming up. I think all of these were represented in the transcript data as well. You'll note that in a lot of cases, the details differ. So the ribosome seems to almost be downregulated, which is a discrepancy we haven't really investigated yet, although I think it's well established at this point that proteomics and RNA-seq don't always accord in terms of the direction. So nevertheless, it's still something that we need to sort of investigate a little bit further. But the takeaway here was that we sort of saw the engagement of very similar pathways as we did in the transcript data. The one piece that I was pretty interested in was the post-translational modification lysine acetylation. So overall, lysine acetylation was pretty heavily upregulated. And there was a pretty heavy involvement for mitochondrial sites, as would be expected. The interesting thing here that we got a little pushback from reviewers on was the expectation that acetylation would be going down because it was assumed that it would be inhibitory in most cases. But this has since been demonstrated in liver in response to CR, in particular in mice, that acetylation levels generally increase and probably related to the availability of acetyl-CoA. But this is also another mechanism that I'm planning to try and investigate further. In the lipidomics data, we saw a couple of interesting trends. The first was in the phospholipids. There was sort of this enrichment for odd chain fatty acids, which was a little bit interesting. There are a couple of potential sources for odd chain lipids, but we don't actually know where these are coming from. So there hasn't been any assessment of the microbiome in these animals, which is one potential source. But they're all housed in pretty much the same set of rooms. So the assumption is that there's not going to be much of a difference there. But this is another thing that will require more of an investigation in an experiment where we can actually test it. Then the other trend in the triglycerides was that there's sort of this bias in favor of an increasing number of double bonds in the chains for the CR animals. And this also was sort of in the opposite direction that I would have predicted, because double bonds would have been a target for lipid peroxidation, which is something that increases with age. But again, here, this is something that's going to take a mechanistic investigation to kind of sort out. And one of the things that I like to highlight with the non-human primate data is heterogeneity and variance. Because they're a heterogeneous model, so none of the animals are genetically the same. And so this is a PCA of the entire lipid profiles for these animals. And the thing that is interesting is you can see that at the 0-year time point, before the animals have been exposed to the diet, they occupy a pretty wide space in terms of variance. The thing that we found with caloric restriction that happens is it tends to compress population variance into a much, much smaller area. And this is true of a number of different features of caloric restriction. And also something that Roz and I are trying to kind of work on in terms of understanding population heterogeneity as a feature of aging. So trying to put a lot of this data together, we generated a very large correlation matrix. So this is basically every molecule against every molecule in a whole bunch of pairwise comparisons. So there's 400 million-ish data points somewhere in this matrix. And then we were hierarchically clustered it to see if there were sort of groupings that fell out of it. And just manual interpretation kind of suggests that there are these 7 big clusters of data. And if you sort of dig into pathway analysis, depending on the data type, you see a lot of the same themes running throughout the clusters. Sort of a lot of the RNA processing stuff seems to be confined down into this cluster. And there's also a ton of small molecules, obviously, that are grouped into some of these clusters. And so the next thing that we're trying to do is interrogate some of these specific relationships between molecules that appear to be moving in the same direction. So that was kind of like a rapid fire run through the liver data. Again, you're going to get tired of hearing me say metabolism, but that is very much one of the common themes. And as I mentioned, the sort of features that popped up that I am really keen to start trying to understand more of is methylation, for one, in terms of its upregulation with CR, and then this RNA processing thing, which it turns out to be, I think, pretty important across the whole network engaged by CR. So one of the other things that we did with this data set is to do a random forest analysis. So as I mentioned, because of the variability of these animals, the traditional statistical testing often leaves us with very little in terms of molecules that are significant. And so random forests kind of takes advantage of the whole data sets. And so we generated random forest decision trees for several of the individual data sets, and then assessed, which molecules are most affecting the accuracy in terms of the ability for the tree to discriminate between the 2 diet groups. And so you get lists like this that tended to be actually different than what traditional differential expression analysis gave us as the significant molecules. And so what I did with those was just, I started putting them into string to see if there was any sort of an association among the factors. And with each type of data on its own, there wasn't really. But if I throw all 3 of them together into string, it produced this nice integrated network that is full of sort of metabolism related things. And then what I did was just ask how much of that network was affected by the RNA processing based regulation that we had seen previously. And it's a really large majority of that. And so we think this sort of suggests that, RNA processing is kind of a key mechanism that is part of how CR exerts its downstream effects. Okay, so the liver, I'm going to skip over that because I kind of already covered a lot of that. The second little vignette that I'm going to talk about is the skeletal muscle biopsies. So in this case, we have 2 groups of animals from which the biopsies were collected. There were very old males, right around their median lifespan, and then late middle-aged males and females, so about 22 to 23 years of age for these animals. And so this study is old enough that the transcriptional analysis was done by microarray. And I will try to make sure I'm very clear on whether I'm talking about microarray or microRNA. But then we also have histological assessments of the tissue itself. So things like mitochondrial activity and contractile area. And so this will probably look pretty similar to what was in the liver. This is just a pathway analysis, and we see a lot of the same themes, mitochondrial energy metabolism, some homeostatic pathways, and then again, kind of down regulation of immune and inflammatory genes. So this is consistent with what we saw in the liver. In the case of the skeletal muscle mitochondria, we can do things like cytochrome C oxidase staining. So this is for complex 4 of the mitochondria. And in the skeletal muscle in particular, there's sort of a distinct organization of mitochondria in terms of mitochondria that are around the perimeter, the subsarcolemmal population, and then the intermyofibular mid-fiber mitochondria. And so what you see is just a 3D rendering of the activity data, activity stain data. And what you can see is that the perimeter is significantly more intense than the mid-fiber. And that's true in the controls as well. And this is this difference in activity between the two populations is something that declines with age. And CR seems to counteract that to a certain degree. So you can see it quantified over here. The gap between perimeter and mid-fiber is there in the controls, but it's substantially larger in the case of the CR animal. Kind of speaks to the metabolic and improved metabolic environment for the restricted animals. And so then that's also just extended to the cellular environment as well. So this is 2-photon measurements of NADH. So looking at fluorescence lifetime and looking at the decay of those photons. And in the control animals, you can see that it's populated with far more red and orange. And then the CR animals, it's much more green, so they kind of have shorter lifetimes, which is influenced by the environment that the fluorophore is in. And this is also, again, a youthful phenotype. In other words, the lifetimes tend to get longer with age, and CR counteracts that. And this is just, so the decay is described by an equation that has a couple of parameters. So mean lifetime is decreased in the control animals, or in the CR animals, I should say. And then A1, which is a coefficient that is roughly the degree to which the cofactor is bound or unbound. And so that's slightly increased in the CR animals. So lipid metabolism has also been changed in these animals. So muscle has a pretty significant store of lipid droplets. And you can see them strikingly in the control animals. You get these really big neutral lipid droplets. In the CR animals, it looks like just a bunch of tiny little blue dots that are actually red. And so the droplets are significantly more numerous in the CR animals, even though the total stain intensity is roughly the same. And the size distribution in the CR animals is skewed towards much, much smaller. And we think this is related to accessibility. In other words, the CR animals are going to lipids much more frequently for energy than the control animals would be. So just back to my harping on kind of variance and heterogeneity. So this is trying to look at a bunch of the systemic data that we have on these animals. So things like body composition, skeletal muscle mass, fasting glucose, etc. With these monkeys, because they are very heterogeneous and we have a relatively small population size, most of these factors are not statistically significant if we just look at them in isolation. But if you look at all of them together in this sort of, it's a type of PCA analysis, you can see that it does a fairly good job of separating the animals by diet. This animal is, as I recall, quite fat, and I don't know what he's doing in the CRs, but considering all of these variables together does a reasonable job of separating the animals based on diet, even if the individual variables don't. Okay, so then the thing that I'm kind of interested in is microRNAs. And so these are, I probably don't have to introduce these too much to this audience. Small RNA molecules that regulate gene expression by participating in the endogenous silencing pathways. And they're interesting for a number of reasons, one of which is they can circulate. And so people are starting to get really interested in them in terms of their potential to serve as a biomarker. But they're also tissue resident microRNAs. And so that's what we're looking at here. And we didn't have an individual microRNA that was statistically significant, but there's a group of them that all come from chromosome 7 that appear to be sort of coordinately downregulated. And a lot of the predicted targets for these microRNAs seem to be metabolism-related genes. So we think this might be an additional mechanism by which CR sort of adjusts the transcriptional program. Okay, so now we're looking at the slightly younger males and females. And so this is back to transcriptional data. And so this is just a volcano plot. You can see the males and females, they don't really have much in the way of the same pattern. And actually the factors that are statistically significant are pretty much not the same at all. And if you can look at them in a string network, see that the functions that they cover tend to be pretty distinct. The females, there seems to be more of an emphasis on mitochondria and energy metabolism, whereas the males are a little bit more structurally focused. It gets really interesting when you look at microRNAs, that same set of groups of microRNAs. Sorry, I realized I should have mentioned that these are groups based on either their sequence similarity or their location in the genome. So these guys on the left, these four are based on sequence, and these are based on location. And in the females, these slightly younger females, we see the same pattern that we did in the really old males. But in the slightly younger males, we see pretty much the opposite. And we don't have a good explanation why this would be. This is, as I mentioned, something that I'm really interested to try and figure out what is going on here. And so we're starting to look at these microRNAs sort of one by one, try and understand their mechanism. Part of the reason for the discrepancy between the males and the females is that they're not really at the same point in terms of the development of like sarcopenia. So if you look at estimated skeletal mass as a function of body weight, you can see that the females there's still a fair bit of overlap in terms of the control and CR group, whereas the males are pretty well separated. And a bunch of the other physiological data suggests that the males are all at this point pretty much well on their way to developing frank sarcopenia, whereas the females really aren't there yet. So the timing of sort of aging in skeletal muscle seems to play an impact. It seems to have an impact in terms of sex dimorphism. But we don't have enough animals in this case to really chase after that. Okay, so basic summary from the muscle stuff. Again, sort of metabolism being one of the themes. And this sort of interesting regulation by microRNAs that may be intersecting with sort of sex dimorphism and how the tissues actually age. Okay, so that was a lot of the work that I did while I was in Roz's lab. And like I said, it sort of identified key mechanisms that we want to start to try and understand what exactly it is that they're doing. So lysine acetylation, microRNA expression, and RNA processing. And so this is just kind of a schematic of the area that I'm really interested in. And it really kind of comes down to gene expression regulation. What is caloric restriction doing, and how is that influencing how is caloric restriction influencing gene expression to then lead to changes in metabolism and lifespan. And so this is from a review article that kind of lays out the landscape of a lot of the signaling pathways that have been implicated in aging. So the sirtuins, AMPK, mTOR, even adiponectin, and the PPARs. And so these are all sort of implicated in aging, and they also are known to be influenced by things like caloric restriction. And the thing that, when I was sort of looking around for good ways to display this, the thing that I was struck by is they all, for the most part, kind of tend to converge on gene expression and the regulation of that process that I think is kind of the interpreter in terms of the signals coming from the environment. And so that's one of the main reasons that I'm very interested in it. And so the other thing that this sort of lays out is a bunch of the pharmacological compounds that can influence some of these pathways. And so those are potential tools we can use to try and dissect some of these mechanisms. And so the rest of my talk, I'm going to talk about a couple of this analysis of data from some mice, one of which uses sort of some pharmacological potential mimics of caloric restriction. And so that was a study that compared caloric restriction to bezafibrates, lithium, and resveratrol. I'll provide a spoiler right now for anyone that is a real big fan of red wine and resveratrol and has followed any of that story. It doesn't do anything. And so in this case, this was looking just at adipose tissue from these animals. And they were on these various treatments for 10 months. And so you can see that just in terms of gross body weight for these animals, again, resveratrol really doesn't do anything. Whereas bezafibrate and lithium both, to some degree, seem to mimic the effects of caloric restriction. And this is true for adiposity as well. You can kind of see the same decline in adiposity.
And what's the target of lithium?
That's a good question. There are quite a few things that it seems to hit. GSK3 beta is probably one of the most well-known and well characterized, I would say. But it kind of seems to do quite a lot of things that we're still don't have necessarily a good handle on. The GSK3 beta, I think, is probably one of the most well-established ones. And so glucoregulation is where the story seems to change a little bit. So this is just looking at fasting glucose, and you can see that now lithium is kind of no longer in the running for being a CR mimetic glucoregulation. And so then we also looked at adipocyte morphology. So this is just looking at the size and distribution of the adipocytes in the adipose tissue. And what you can kind of see here is that, you know, CR and bezafibrates kind of shift things towards smaller adipocytes. This is kind of analogous to the lipid droplets in the skeletal muscle. Lithium really shoves things to a much smaller adipocyte size. And so you have way more numerous adipocytes and they're a lot smaller. If we look at the transcriptional data for these animals, this is where the volcano plots actually seem to have similar patterns. And I've dropped resveratrol out because, like I said, it does nothing. There were 2 transcripts, I think, that were statistically significant. Sorry, say that again. Oh, the food intake was monitored for all of these animals. So yeah, they were eating a defined amount of food in all cases. Oh, so that is a good question. And I actually don't know, that decision predates my involvement in the study, so I actually don't know the answer to that, unfortunately. There's literature on several of these. So bezafibrate has been used clinically, as lithium has as well, actually. And so I believe it was probably based on literature in terms of the dosing.
Patients with bipolar can be on lithium
Mhm.
for a really long time. Other eventually damage to their kidneys, but is there any evidence in humans for lithium that's
Not that I'm aware of. I don't know that anyone has really looked at sort of what lithium might be doing metabolically for a lot of those individuals. It's sort of anecdotally, I think the issues with glucoregulation are a known side effect with extended lithium treatments. But beyond that, I'm not aware of any studies that have really examined if lithium might look like some sort of food restriction in humans. Right. So when it came to the overlap of the significant factors, there actually was a pretty decent amount of overlap in terms of the three treatments kind of hitting some of the same things. There's actually fairly good overlap just between bezafibrate and CR as well, although in each case we get independent things that are changing in response. The things that are shared in common are pretty much all moving in the same direction, with the exception of whatever this guy is right here. So it appears to be doing kind of the same thing to the things that are shared. And at the pathway level, maybe not surprisingly, there's quite a lot of overlap, and again, very similar similar pathways to what we saw in the CR data. When it comes to the RNA processing story, this is where, again, bezafibrate seems to be very, very similar to caloric restriction. Lithium and really sort of induced a lot of changes to exon usage. And this is maybe not surprising given the number of things that it appears to affect. But we haven't yet dug into kind of what is going on here, although we have plans to do so. There's not quite as much overlap in terms of which things are changing in this case. Yeah. Adipose. This is adipose, yeah. And yet again, resveratrol does nothing. And so, quick summary for this: one of the takeaways is basically bezafibrate seems to be a pretty decent CR mimetic, at least from an adipose tissue perspective. Although obviously it would still require quite a lot of testing to figure out if it really does a good job. Lithium seems to be similar to CR in a number of respects. And I'm interested in lithium because it seems to really activate this RNA processing phenotype that we've seen. And so lithium is maybe a potential tool that we can use in things like cell culture to try and start understanding what exactly it is doing. Okay, so then the final story that I'm going to talk about a little bit, and one that's ongoing, part of the work is being done actually in my lab, and Spencer is doing some of it, is to look at just a caloric restriction and aging in mouse brain, actually. So these were animals that were started at 2 months of age. They were maintained on the diet until defined time points of 10, 20, and 30 months of age. So, you know, reasonably youngish mice, middle-aged mice, and very, very old mice. And if we look at PCA, the transcriptome, for these animals, the CRs cluster altogether. And then the controls are kind of spread out more along the other side of the graph. So in the first dimension, it kind of seems to separate pretty well. And if we look at the volcano plots for these, so I was pretty conservative in terms of what I decided was statistically significant. So these required a log 2-fold change of 1 or higher, and FDR of less than 0.01. So there's quite a lot of stuff changing in these animals. But a lot of it doesn't really seem to overlap across the 3 different regions. So in terms of the factors that are significant, there's only about 75 that are shared in terms of the response to caloric restriction at those 3 ages. And there really was very little overlap in terms of the pathway. So this is something that we're still trying to figure out, because I think our expectation would have been that they should have been fairly similar things changing at all 3 ages. So with this project, we also collected lipidomics from basically the same samples. And so this was performed by the Biotech Center here. And so we're sort of in early days with this, but there are a lot of things changing in response to CR, or excuse me, in response to age, I should say, that is pretty heavily influenced by ceramides. There are a lot of ceramide species that are changing in response to, so this is the 30-month versus the 10-month comparison. And so if you look at specific classes of these lipids, you can see that there are some rather dramatic changes in the control animals that are not necessarily reflected in the restricted animals. And so this is sort of a correlation matrix from all of the lipids. And so just looking at a couple of these clusters that fell out of that, the changes as a group seem to be kind of subtle, but we seem to see some slight changes in the restricted animals in particular across these 2 going up versus this cluster where they're going down. And so what we know so far is that this cluster seems to be enriched with docosahexaenoic acid species, whereas this one seems to be biased in favor of fewer unsaturations. So these are things that we're going to be investigating more specifically. And then one of the things that Spencer's been doing is trying to look at integrating these 2 data sets together. So first sort of attempt at that was this very big correlation matrix that includes both the lipids and the transcriptional data. And so this is filtered just based on whether the correlations were significant or not. And you can see we kind of have basically 2 big clusters that are anti-correlated to one another. So we'll be dissecting the relationship of the molecules in those clusters to try and understand what their mechanistic basis is. So that is sort of where I'm going to stop. Like I said, the future directions for a lot of this stuff, trying to integrate. So this is specifically for that project to integrate the transcript and lipid data a little bit more. In the case of that project, we also have the RNA-seq data is quite deep, went for about 100 million reads per sample. And so the hope is that we can start to try and assess RNA processing really well in that data set. That data set also, unfortunately, is only male mice. And so we will need to do females as well at some point. And then based on the monkey work, my lab is also generating some CRISPR cell line and specific factors to try to tease out a mechanistic basis for things that we observed there. So with that, I will thank both Leigh and Spencer. My start of my lab, obviously, a lot relies on my time in Roz's lab, and that's not even all the people. So there's not enough room for them. And then one of the things I always like to highlight with the monkey story in particular is that's a huge collaboration across a lot of groups on campus, and our ability to do something like that is heavily dependent on a lot of great collaborators here at UW-Madison, including Ricky Coleman, Josh Kuhn, John Danu, people here in the Biotech Center as well. And then very appreciative, of course, of funding for myself from the NIH, and then also Roz's program has a funding from the VA, and then a pilot award from the Conference of Entities. And with that, I will stop.
So what about in your primate, in a female primate with menopause or restriction and sort of changes to that? Obviously, you saw some differences in the sexual dimorphism, but maybe talk a little bit about differences there.
Yeah, so as far as I know, Ricky has not looked too much at that. So the animals, rhesus monkeys, they do go through menopause, but it's, I think, much later than would be corresponding in humans. So they generally maintain estrus for much later. And so I don't know how much she's looked at. You know, there'd only be 15 female restricted monkeys, and I don't know how many of them lived long enough to make an assessment for that. It's something that is known from other organisms that's going to, in the short term, CR would impact something like fecundity and reduce it, potentially extending the amount of time that individual is fertile though later on. So, saw another.
Yeah, I was wondering, in your you mentioned the CRISPR cell lines as what you're seeing, but you're seeing like a lot.
Yes.
I'm just wondering, like that was big. I was just wondering, what are you excited about following up on it and why?
Right. So the thing that I got most excited about, it sort of ties together two of the pieces that I'm really interested in. So one of the things with the acetylation that we observed, there were big changes to acetylation on a bunch of RNA helicases, which would be RNA processing enzymes. And so that's where we're starting, is making some mutants of those sites, also just sort of knockouts of those enzymes, and trying to understand if there's a connection between the site of acetylation and RNA processing. So that's the main one that we're trying to get started.
And also, I was just curious because that's not the model I work with, but I was just wondering, like, if you figure out the mechanism in the lines, right? Like, is the idea that there was some drugs or something that manipulates you would go back to the monkeys and then treat them?
Probably not going back to the monkeys, but mice would certainly be on the table.
So what? But you did the monkeys, some things could be different monkeys and
The idea with the monkeys is that so they are highly translatable and presumably most relevant for humans, but doing any sort of a mechanistic intervention in monkeys is really challenging. unless we were at a point where we were pretty sure we had a drug that was working. Would go to cells and then mice to work out mechanism. Maybe monkey experiments in the future for that. Again, if we're really sure there's something that would work. But I think the cells and the mice are going to be important for actually working out mechanism.
I don't have a follow-up on that. So how do you verify that the methylation and RNA processing observations hold in mice?
Yes, it's not exactly the same, but and we haven't confirmed necessarily like the individual factors from the monkeys, but CR does induce sort of RNA processing in a number of different tissues in mice. And the couple of HeLa cases that I'm looking at are pretty much completely conserved as well. So that's, we think we're okay there.
Two quick questions if possible. I can also do one. My first one is in caloric restriction experiments, how sticky is the change? So if you calorically restrict an animal for 10, 20 months, and then you let it eat ad libitum, are these plastic responses? What's their delay in returning? And could that give you insight into long-term changes versus, you know, like your temporal experiment? Any thought on that?
Yeah, so it actually, okay, I won't go find it. I had it on one of my slides as one of the questions, because that actually is something that I have spent a lot of time thinking about, is the persistence of these events. I think the answer is that there is likely some persistence, but exactly how much, I don't think we know that yet. So it's a hard question to answer. But it is something that I think you could start to try and address it with some different timings in mice. So putting them on short-term and long-term CR at different time points, and looking ultimately at longevity.
One more real quick, just on the skeletal muscle dimorphism, skeletal muscle. I'm not in your field. I work tangentially with metabolism, but was that was a shocking amount of dimorphism. There's like no overlap. Is that expected? Is it because you chose a tissue, for example, when I think of skeletal muscle, it's one of the first things I think of in terms of dimorphism. So males hormonally will put on more muscle, you know, in a going from adolescent to puberty to adult. Do you think if you chose a different tissue, you would see more overlap? And then my last comment is just, does that suggest that aging is vastly different in males and females? And what do you think about that?
So I think it's probably, well, to answer that question first, I think it's probably accepted that aging in males and females is different at some level. I think you're right in that skeletal muscle is going to be one of those tissues where the difference is going to be starker, just from a pure body size standpoint. So in the rhesus monkeys, the dimorphism in terms of the size of the animals is roughly what it would be in humans. And so I don't know that I would expect that much of a difference in other tissues necessarily. You know, a lot of the phenotypes that we see of aging are consistent in males and females, even if some of the smaller details do vary. So it's something that I think just sort of reinforces the need to get more data. You know, in a lot of these biomedical experiments in history, we didn't use females. And I think that's something that needs to be rectified going forward.
Yeah. Thank you.
Yep.
Great talk. I had a question about, you mentioned that there's a lot of differences in lysine affiliation for various proteins. But aging and diet have both been shown to have major impacts on like epigenetic modifications based on metabolite availability. So I was wondering if you're thinking about looking into that.
So yeah, that's certainly on the radar screen. I will say that for the most part, we didn't capture histone acetylation. So that is mostly non-histone acetylation, which is the piece that I think I'm a little bit more interested in. we've got a couple people on this campus who are very, very good at the histone acetylation in particular. But yes, certainly in something like liver, I think the expectation is there's going to be some significant dynamics there related to dietary intake. But that's something that we'll have to kind of work out in mice. Yep.
How do we do, a little bit more by they are made if they had something that started on the ground. We could add, phenotypes.
Yeah, so basically the way that's assessed is from short read RNA-seq data, and all it's basically doing is looking at the number of reads within an exon and comparing it across the two diet contrasts or across the two diets. And so that can arise from a number of different mechanisms. So alternative splicing being one, alternative start sites or polyadenylation are also potentially can give rise to that. So in our data, we don't really know yet specifically what is occurring. So that's something that we still have to investigate more specifically.
It's just like it's a sequence present RNA.
It's just a difference in terms of the reads that go through that sequence. So for the exon, and presumably that means that there's more or less usage of that exon, basically. Does that answer your question?
Well, it's just, I mean, it's not clear to me that it's taking a relative measure, or there are segments of genes that are where there are no needs that cover that segment. There's explicitly no reason that this cohort that does.
Oh, so I would say that for the most part, in all of the cases that we have in there, there are reads that go through that spot in both diets. It's just the degree changes in response to the diet. So I don't think we've uncovered any sort of alternative, like cryptic exon or anything like that, as far as I'm aware.
Do you have any, like, I'm curious to see more of the
I
think it's the same thing. I mean, do you ever, I mean, I think, it sounds to me like the data could be suggesting that sometimes those exons are included in, in some of the characters, sometimes not, right? So in theory, you should be able to find breeds that are, they go through those alternate exon junctions. Right, so in theory, somewhere in that pile of reads are a subset of it, I don't know, a junction between exon 1 and 3, as opposed to 1 and 2, or 2 and 3, right?
yes,
Have you spent any time looking at that side of something?
we've done a little bit. That's obviously sort of one of the things that it's top of the list in terms of things to do. It was one of the things that the reviewer asked about in the paper, and so we provided a couple of examples. But I wouldn't say that we've gone in and made any sort of comprehensive assessment yet.
For a short range, it can be hard to get a top.
That's actually one of the things that I have planned is to do some long read sequencing, both for the CRISPR lines and then potentially some of the mouse tissues if we can get it. The monkey tissue is a little bit more difficult. But yeah, long read sequencing is one of the things that I want to explore to try to answer that question.
Not clear.
Yes. Yeah.
Why do you correctly restrict function?
Not very easily.
Is that because they can die, or because like caloric restriction is maybe changing the whole organism through the liver first, and then the liver signal?
There's certainly some systemic stuff at play, but I think just in terms of practically speaking in cells, we cell culture generally sort of bathe the cell in glucose, and the degree to which we can ratchet that down, I think is a bit challenging for actually inducing a caloric restriction phenotype in cells. So in our case, the cells are sort of something that we can test specific factors in, using inhibitors or drug treatments or siRNA or things like that. I'm not planning to try and do CR in a cell culture dish, because I'm not sure what that would look like.
Okay, we have no more questions. We'll say thanks again.